Hello, everyone. My name is Megan, and I'm going to be presenting to you the paper that I chose, which is titled High Resolution Detection of Translocation of Oral Bacteria to the Gut. So, a little bit of background information is that the oral cavity has its own community of microbes that are distinct from the gut. It provides access to the gut via the digestive tract and is the second largest diverse microbiota after the gut. The oral cavity contains over 770 species of bacteria, and the reason for this diverse environment within the oral cavity is from the different microbial habitats, such as your teeth, your soft and hard palate, and your tongue. The gut contains microbes that can prevent the colonization of endogenous microbes. However, this isn't always the case. So, ectopic colonization of microbes from the oral cavity into the gut refers to the presence of oral microbes in the GI tract, particularly in areas of the gut where they are not typically found. And there are two proposed routes of how oral microbes can be translocated into the gut. First is hematogenous route, which involves microbes from the oral cavity disseminating via the bloodstream. So oral microbes can get into the blood via the bloodstream from injuries inside the mouth, such as if you're brushing too hard, as well as or from periodontal disease like oral cancer or periodontitis. The second route is enteral route, and this is when oral microbes can disseminate from swallowing. Normally, a healthy gut can handle oral microbes, and it can prevent colonization because it has certain microbes and mechanisms to prevent such colonization, such as your gastric acids. Most bacteria from the oral cavity are very sensitive to the gastric acids in the gut. But when the gut is in a state of dysbiosis, this is where you can have problems arise from oral bacteria getting into your gut. The health implications that ectopic colonization of oral microbes into the ectopic colonization of more oral microbes have when they get into the gut are good and bad implications. So one of the good implication is of species Veillonella, which is a species of a certain bacteria found in the oral cavity that can modulate immunity in the host. So when you have ectopic colonization of this type of oral bacteria into the gut, it can actually help in gut immunity. Also, there are species of Klebsiella isolated from the oral cavity that are capable of inducing T helper 1 cells once they're colonized in the gut. However, Klebsiella species have disease implications such as in IBD, where when the gut is in a state of dysbiosis, species of Klebsiella can cause major inflammation in the gut. Also, another disease implication is IBS. In individuals with IBS, there are certain species of oral microbes that have been found in the gut, such as Streptococcus species. And lastly, colorectal cancer. So patients who have been diagnosed with colorectal cancer have an increased rate of transmission of microbes from the oral cavity to the gut in comparison to healthy individuals. To get into the why of this paper, there isn't sufficient evidence regarding the transmission of the oral microbiome to the gut microbiome. And some of the questions that the authors aim to kind of answer in this paper is is there a direct connection between the oral and the gut microbiome? And what are the contributing factors that are involved in the increased presence of oral microbes in the gut microbiome? And answering these questions are important because it can provide a better understanding of the relationship between the oral and gut microbiota, as well as diseases related to the gut microbiota. So now I'm going to get into the methods of the paper. So the authors gathered 144 pairs of stool and saliva samples from adults ranging from the ages of 40 to 80 years old in a community in Hisayama Town, Japan. And they obtained oral and just general health information from all the participants. And they extracted DNA from both saliva and stool samples. So they extracted DNA from the saliva samples using the bead beading method, where they just used beads basically to break up the saliva samples. In the stool samples, they extracted DNA using an automated DNA isolation method, which is just basically a machine that does everything for you. There's no manual work involved, followed by the beading method. And then they use PacBio sequence or PacBio to sequencing of the 16S rRNA gene amplicon. And then they use an RDP classifier, which stands for Ribosomal Database Project. And this is just a database that's based on 16S rRNA sequences from bacteria, archaea, and fungi. And they use this to classify each of the sequence variants. So another thing that they also used is called amplicon sequence variants, or ASVs, which is an alternative method to OTUs, or the operational taxonomic units. And they just used amplicon sequence variants to examine microbial communities. Then they use BLAST for species level taxonomic assignment, and then they use various statistical tests to analyze the data. So one of the tests, or one of the statistical methods that they used, was a Bray-Curtis index, which they use this to evaluate the differences in the bacterial composition between the salivary versus the gut microbiota. Then they use a Crystal Wallace test to show whether the total abundance of the specific ASVs that were found in several of the study subjects' gut microbiome varied depending on different clinical factors. Then they used an LDA, which is a linear discriminant analysis. So they used LEF SE, so it's an LDA with effect size. And this is a tool that's specifically used in researching the microbiome. And it identifies microbial features that are significantly associated with specific groups or conditions. Lastly, they use a Shannon diversity index, basically to just measure the diversity of species in a community. I'm going to get into the results of the study. So, this first figure is a comparison of the microbes found in saliva and stool samples. So, figure A is a plot of the saliva and stool samples where they use the Bray-Curtis method to measure the abundance of the ASVs. And what figure A is showing is that the microbes from the saliva and the stool samples are very different. Then in figure B, it's a phylogenetic tree and is showing the relative abundance of each genus that was found in the saliva and stool samples, where the most abundant genre in the gut was Bacteroides and Faecialobacterium. And then within the saliva samples, the most abundant genre was Neisseria and Streptococcus. When looking at both the saliva and stool samples, the most abundant genre was Prevotella and Streptococcus. And they did find that there was a slight overlapping of two genres, Veillonella and Megasphaera, in both the saliva and the stool samples. For figure C, is showing the distance between each participant's saliva and gut microbial communities. So around 27% of the subjects, so 39 out of the total number of subjects, this distance between their saliva and gut microbial communities was 1.0, which suggests that there was no overlap or sharing of ASVs between their saliva and gut microbiota. So their saliva and gut microbiota had completely different microbial compositions. In comparison to the remaining about 73% of the subjects, which was 105 out of the total number of subjects, the distance between their saliva and microbial communities was less than 1.0, which shows that there is some degree of sharing or similarity of the ASVs between their saliva and gut microbiota. Basically showing that, or indicating at least one ASV was found to be common between their saliva and gut microbiome, suggesting that there is some level of overlap in microbial composition. So in this figure, figure 3 is showing the extent of microbial sharing between the salivary and gut microbiota. So figure A is an extension of the previous figure with figure C, where it's showing the total relative abundance of shared ASVs in the gut microbiota, basically suggesting that there is a significant portion of the participants that had some degree of similarity or sharing between their saliva and gut. Although the abundance of the shared ASVs differed among individuals. In figure B is showing the predominant shared ASVs. So the most frequently shared ASVs found in the gut belong to 3 genres: Streptococcus salivarius, Streptococcus parasanguineus, and then Veillonella dispar, where these specific microbial genre were the most common uh, in both the saliva and the gut of the participants of this study. Figure C is showing the transfer ratio and the frequency of the shared ASVs. So the transfer ratio was used to measure the frequency of ASV sharing between the saliva and the gut for different microbial genera. And what they found was that Streptococcus had the highest transfer ratio, suggesting that this genre was frequently shared between the saliva and the gut. Whereas some genre, such as Prevotella and Megasphaera, had a transfer ratio of 0, indicating that there was no detectable sharing of ASVs between the saliva and the gut. There were other species levels, or excuse me, similarly, at the species level, the transfer ratio of Streptococcus salivarius and Streptococcus paraguineus was around 0.58 and 0.27, suggesting that there was frequent sharing ASVs. So in this figure, or a figure 4, is showing factors influencing the increased presence of oral microbes in the gut. So figure A is showing the clinical factors that affect the relative abundance of the shared ASVs in the gut microbiota. And what was found is that age and dental plaque had a significant effect on the relative abundance of ASVs, of the shared ASVs. And they used the Kritzgall-Wallace test to determine this. The other clinical factors, such as BMI, drinking, sex, didn't have any significant effect on the relative abundance of the shared ASVs. In figure B, figure B is showing the significance of dental plaques impact on the relative abundance of shared ASVs in the gut, where individuals who had moderate amounts of dental plaque accumulation had a increased or higher abundance of ASVs in their gut microbiota compared to those who had no dental plaque. Figure B also shows that with age, there is an increase of these shared ASVs in the gut. So, for example, with the 80-year-old participants, they had a significantly higher abundance of the shared ASVs when compared to like the 40 to 50-year-old participants. So in this last figure, figure 5, is showing the relationship between the shared ASVs from the salivary and gut microbiota. So figure A is where researchers use the LDA, the linear discriminant analysis, or the LEFSE, with effect size, to identify the microbial genre that could be effectively distinguished between two distinct groups of subjects. And what they found was that there were 7 genera that were significantly associated within these two groups, and suggests that these particular genre were distinctive, and played a role in differentiating between the two groups. Figure B is showing the results, or excuse me, Figure B, the results provides specific information about the differences in the relative abundances of certain microbial genera in the gut microbiota between subjects with higher and lower shared ASVs. So subjects or participants with higher shared ASVs in the gut had a significant increase in the relative abundance of bacteria such as Streptococcus, Lactobacillus, and Klebsiella. Whereas the relative abundances of bacteria such as Faecilibacterium, Blachia, Megamonas, and Parabacteroides were significantly lower in the gut of subjects that had higher shared ASVs. Some of the key findings of what the researchers found from the data was that there's extensive translocation of oral bacteria to the gut. So ASV shared with the individual's own salivary microbiota were detected in the gut microbiota of 72 to 73% of the subjects. That was the 105 out of the total number of subjects. The abundance of these shared ASVs in the gut varied, ranging from about 0% to around 63% of the gut microbiota for each of the subjects, which suggests that the oral bacteria are extensively translocated into the gut in community dwelling adults. Secondly, age and oral microbiota individuality. So the study found that the oral to gut microbial sharing and the composition similarity were higher within an individual compared to those across the different individuals, suggesting that the oral bacteria found in the gut tend to be more similar to the individual's own oral microbiota. Lastly, associations with the composition of the gut microbiota. So the study observed that individuals with a higher abundance of shared ASVs in the gut had lower levels of beneficial gut commensal bacteria, such as like certain pathogenic bacteria like Klebsiella pneumoniae, which has been known to cause inflammation in the gut. So was significantly more abundant in the gut of individuals with an enrichment of oral bacteria. So all of these findings suggest essentially that the translocation of oral bacteria into the gut is a common events in adults, and it may be influenced by factors such as age. However, the presence of oral bacteria in the gut was associated with a decrease in beneficial gut bacteria and an increase in potentially harmful or pathogenic bacteria. Kind of talk a little bit about some limitations of the study. The first is that the saliva and stool samples were taken within 1 year of each other. So there was a time gap between the sampling collection where the saliva samples were taken in 2017 and then the stool samples were taken in 2018. They didn't really discuss why there was this time gap. But typically, the microbial community within the salivary microbiota in humans is stable over time. However, the microbial composition in the saliva could vary annually, and this could affect the results. And by underestimating the number of ASVs in the gut. Second, there is a differences in read numbers. So differences in the number of DNA sequencing reads gathered from each sample could introduce a bias and underestimate the microbial sharing. Third is sampling bias. The study only considered age, which could create bias since other factors that might influence the relationship between the oral and gut microbiota since there are other factors that could influence the relationship between the oral and gut microbiota. However, this study did show that there wasn't any significant differences in the oral and the general health conditions between included and excluded study subjects. Next, 16S RNA gene analysis. So this technological approach doesn't really consider the activity or the viability of these organisms. So ingestion of salivary bacteria or microbes from your oral cavity isn't sufficient to account for their significant presence in the gut. Instead, it's just suggested that these bacteria are actively growing and replicating within the gut environment. But this paper doesn't really focus on the underlying mechanisms. So the study only identifies the association between the abundance of oral bacteria that's found in the gut, and it doesn't really explain the mechanisms that are involved in this association. That I personally had, and maybe this could be part of the discussion between everyone, is why isn't there enough or sufficient evidence on this particular topic of the ectopic colonization of oral microbes to the gut? And is the evidence not sufficient regarding this particular topic, or is there not enough evidence or not sufficient evidence in regarding the microbiome in general? Also, what is the correlation between age and dental plaques? Why why is it that age and regarding an 80-year-old having more dental plaques in that association with ectopic colonization of the oral microbes into the gut. What are the actual mechanisms that drive the association between the ectopic colonization of oral microbes into the gut? And what is the current research on this topic? Has there been any new developments or progress? Is this like a newer subject, something that's newer in the area of study of the microbiome? Thank you for taking the time to listen to my presentation. And these are just all my references that I used. Thank you.